Let's get started here. So welcome everyone to this month's COVID Immunology Consortium Symposium. Delighted to introduce two fabulous speakers for you today. Before we get started, just a land acknowledgement to say that BC Children's Hospital Research Institute, where I'm based, operates on the traditional ancestral and unceded territory of the Coast Salish peoples, including the Musqueam, Squamish, and Tsleil-Waututh nations. And I would like to extend this acknowledgement, gratitude, and respect to all Indigenous peoples on whose traditional territories we work, learn, and build on. So just for people who are new to the group, the COVID Immunology Consortium-BC, or CIC-BC, aims to bring together researchers at all stages of their career to help facilitate collaborations with people interested in infectious disease immunology. We have over 100 researchers across BC who are a member of the consortium, and we communicate with quarterly newsletters, bi-monthly research symposia such as this, and we have a very active and engaged student and trainee group as well. And there's a QR code there that links to the survey, which will help us understand your research interests, and how we can improve the consortium to better serve the community. And then I can't see the chat right now, but I think Beth will also put that link to that survey in the chat. So today I'm delighted to welcome Dr. Peter Brodin and Fatima Yaseen as well to speak at the symposium. So I'm going to start off with Dr. Brodin. So he is the Garfield, Weston Chair and Professor of Pediatric Immunology at Imperial College London and Professor of Pediatric Immunology at the Karolinska Institute in Stockholm, Sweden. After completing his MD PhD program at the Karolinska, Dr. Brodin joined Mark Davis's laboratory at Stanford as a postdoctoral fellow investigating the contribution of heritable and non-heritable sources of variation in immune systems of twins. He then returned to Sweden to establish a national facility for immunomonitoring at the Swedish Infrastructure Lab, Science for Life Laboratory, and his own research program applying systems immunology to study of the immune system development early in life. His lab develops and applies novel experimental and computational methods to describe human immune system variation, with a particular interest in the immune systems of children, early childhood development, and role in health and disease. So Peter, I'm happy to do your slides for you if you prefer, or I can hand it over to you if you'd rather share. I don't mind.
I can control them and share myself if you don't mind.
Sure, yeah, that's fine. I will stop my sharing, and then yeah, you can do that.
So that should work, I hope. Otherwise, please.
No, it looks great.
Okay, so thanks so much, Manish, for the invitation, you and Beth, and also that kind introduction. I'm really happy to speak to you today as a sort of pediatrician and particularly immunology interested. I've been working quite heavily during this pandemic with a number of things related to COVID-19 in children. And so I'm going to tell you a little bit about what we've learned, not just from my own work, but also from collaborations with many others and also the work of other people. So I'll start by just telling you, as you probably already know, that this one virus, new to all of us, obviously has led to both mild and severe COVID-19 disease. And a lot of work has gone into try to understand what determines that. And I think we have learned quite a bit. And I'll talk a little bit about that. And then obviously multisystem inflammatory syndrome is something that I have been very actively involved in, both as a clinician and research. I'll touch upon this severe acute hepatitis of unknown origin and present the hypothesis that I have been working on together with colleagues, and which is still just a hypothesis, but I still wanted to highlight it. And then I'll end by saying a few words about long COVID, which is work in progress from our group. So we are members of the COVID Human Genetic Effort, which has been a phenomenal group to be a part of during the pandemic, where As you can see, people from all over the world, including UBC, have been involved in working to try to understand exactly this that I mentioned, why with this novel infection, some individuals develop unexpectedly severe disease, for example, young children with life-threatening COVID pneumonia, but also some subgroups within the consortia are trying to understand how some individuals could have some form of resistance and unexpectedly prevented from being infected, or at least develop severe disease despite having old age and so on. And as the pandemic has rolled on, this group has addressed new challenges such as MIS-C, such as vaccine side effects, long COVID, and so on. And so it's been really phenomenal to be part of this collaborative group. So when it comes to mild versus severe COVID, most of these studies have been done in adults, obviously, because they are the ones that typically end up in intensive care units with acute COVID. And by sequencing whole genomes of individuals under 50 years of age, so sort of on the younger spectrum, who developed life-threatening SARS-CoV-2 induced pneumonia, the groups of Jean-Laurent Casanova and several other collaborators within this network, including our group, were able to find rare variants in sensing molecules such as TLR3 and 7, or signaling pathways downstream of these that could impair the induction of type 1 interferons and leading to impaired responses by respiratory epithelial cells to SARS-CoV-2 infection. And that could explain 5% or so of these individuals with life-threatening pneumonia under 50 years of age. A phenocopy of this was found to be neutralizing autoantibodies to interferon, all of the alpha species and also omega. Curiously, no autoantibodies neutralizing beta interferon. And this was intriguing, and that this could explain actually a lot more, almost up to 20% of severe life-threatening pneumonia in patients infected with SARS-CoV-2. And even more curious, to me at least, was the fact that these autoantibodies seems to be increasingly present with advancing age. And they are more present in males than in females, which we still don't understand why. But these two sort of lines of evidence basically suggest that type 1 interferons play a very critical role in the defense against SARS-CoV-2 infection. And this is further illustrated here. So this is a child who I was peripherally involved in treating. This child had critical COVID-19, was on a ventilator, and this patient had an underlying condition called APS1, or autoimmune polyglandular syndrome, which is a deficiency of the gene AIR, an important regulator of central tolerance in the thymus. These patients have been known for a long time to have a high propensity, or almost more or less all of them have high levels of neutralizing autoantibodies to type 1 interferon. And this was basically the inspiration for those studies that I mentioned previously. And what happens here is this particular child came in, required a ventilator, was very sick, and was then treated by plasmapheresis. And what you can see here is these gray bars represent the different sessions of plasmapheresis leading to reduction in procalcitonin, gamma interferon, CRP, and an increased CT value, reflecting a reduction in viral load. So this patient recovered as a consequence of this treatment, further illustrating the importance of type 1 interferons, particularly early in the course of disease, and pointing to the role of these autoantibodies. But if we get back to this topic, which I started out by saying, namely why COVID-19 is generally so mild in children as compared to older people. One thing that's really curious is that in this case shown by Sam Palmer and colleagues, the rate of hospitalization shows a very gradual increase with time. And so it's quite intriguing that there's such a gradual increase. And this has been reproduced in many different countries, as you can see, suggesting that there's something in the underlying biology, which is also probably gradual over time, that could explain this difference, with some exception here very early on. And so one of those things could, in theory, be thymic involution. We know that the immune system of young children are basically designed to meet new challenges with a very effective thymus that outputs new T cells all the time in order to face new challenges and develop memory responses. Whilst an adult individual more dependent upon memory recall responses. So this gradual rate of increased hospitalization could to some extent fit with the gradual decline or involution of the thymus. So what Sam Palmer and colleagues at Oxford did was they modeled the rate of hospitalization and in relation to the rate of thymic decline, which is well known and shown in previous studies. And when they did that, they found that there was about 50% of unexplained protection from severe disease in the young individuals under 20 years of age, meaning 50% of their sort of propensity to have mild COVID could not be explained by thymic involution alone, and some other factor must explain this. So what might those other factors be in individuals under 20 years of age? Well, a number of different hypotheses have been brought forward. Cross-protective immunity from related viruses is one possibility, and I think there is now some literature support that this might be the case. Probably not the entire explanation, because why would that be such a gradually increasing or decreasing phenomenon? I don't really see why that would be. Viral interference is another issue where particular viruses could interfere with SARS-CoV-2 in the nasal mucosa and oral mucosa, preventing infection or leading to milder infection. Again, very difficult in my view to explain the gradual increase in hospitalization with these factors. What has been more clear though is that there are local tissue differences in the way that the tissue is arranged and organized, and the way that the immune cells in the tissue responds to stimuli in children versus adults. And this is a summary slide showing a number of studies that have contributed to this, basically saying that immune cells in the nasal mucosa and oral mucosa is preloaded and ready to go with type 1 interferon responses, as well as the epithelium upon infection in children, which is different from that of adults. There are also differences in the immune cells that are present in the mucosa prior to infection in children versus adults. So this is probably one important factor. All right, so this is kind of where we are, I think, in terms of mild to severe COVID. I will present another hypothesis that I have brought forward towards the end of the MIS-C section here, because I think they are tied together. But MIS-C then, obviously for many of you, I think this is quite evident by now that there is some overlap in presentation with Kawasaki disease. There's also some overlap with toxic shock syndrome. However, the disease seems to be a unique entity, different from either of these conditions. When we started seeing these patients in the spring of 2020 in Europe, we could not really tell them apart. There were some curious features, such as these patients typically being a little bit older than our classical Kawasaki disease cases. They were more severe than most of our Kawasaki disease patients. Many of these patients had shock, required intensive care, and in at least in the UK and the US, there's been a number of deaths. So what is this? We began by studying the very first cases that we could sample in Sweden and in Italy, together with colleagues there. We compared these individuals with respect to plasma cytokines, immune cell activation, antibodies to SARS-CoV-2, but also to a number of other viruses. We compared Kawasaki disease children sampled prior to the pandemic, healthy children, and then also SARS-CoV-2 infected children. And what we found, since this is published a while ago now, to summarize what we found basically was that the cytokine storm in MIS-C is quite unique from that of acute COVID, with severe patients, typically adults on ICU and so on. So there's a cytokine storm, but it's a very different type. They're different cytokines and compared to acute COVID. There's also clear differences between MIS-C and Kawasaki disease here. There are 3 patients that overlap with the Kawasaki group, and we think those are actually misdiagnosed patients with more classic Kawasaki. We found also in this study published in the, I think, late summer or September of 2020, that there were autoantibody profiles in these patients with MIS-C that were characterized by very broadly reactive autoantibodies. So it's very different from, let's say, Addison's disease or autoimmune thyroiditis, which are classical autoantibody mediated diseases, where you have one specific target and very high titers of autoantibodies. In this case, what we found was basically a broadly reactive autoantibodies against a number of different antigens. And this was found also by other studies from other groups who also found autoantibodies in MIS-C patients, but not one specific antigen target. It's important to note, because some people have raised the concern that our findings were confounded by IVIG, which can obviously carry autoantibodies. But it's important that all of this data was collected, all of the samples were collected prior to IVIG treatment. And clearly, I think this indicates that there is an element of a loss of B-cell tolerance in MIS-C, and autoantibodies probably explain part of what we see. One example, which from a recent paper, shows autoantibodies against IL-1RA, which is basically the endogenous inhibitor of IL-1. And we give this in Sweden. This was the most common treatment for patients with severe MIS-C in the form of the drug anakinra, or IL-1RA recombinant. And so this was interesting to us, that some people claim that autoantibodies to IL-1RA could be explaining some of the pathology. So we tested that in our setting using 54 patients with MIS-C and their plasma samples. Here you see autoantibodies to type 1 interferon, and to the far left are patients with APS1, which I mentioned earlier. All of them have very high titers of autoantibodies to type 1 interferon. And here is IL-1RA or IL-1RN. And as you can see, there is a little bit of reactivity in the MIS-C patients, with one exception here with very high activity. But for the most part, we see that reactivity among healthy children, and also look at this IVIG prep here with sort of higher titers. And so I personally do not really believe that IL-1RA autoantibodies plays a major role in MIS-C, but this remains to be seen. It's very early days, I think. I also think those patients should not respond so effectively to anakinra if they had such autoantibodies. What is quite clear, though, from a number of studies, is that there is support for super antigen mediated T cell activation in MIS-C. And for those of you who are not familiar with a super antigen, basically what this is a protein or a part of a protein that is able to, in a non-specific way, activate large numbers of T cells. And they do so by binding on the side of the T cell receptor together with the MHC and activating T cells that carry a particular V beta chain, even though the alpha chain might be very diverse. So this can lead to up to 20% or so of the T cell receptor, T cells actually, to be activated. And we can see this by looking at T cell receptors repertoires, which are skewed towards one particular V beta, in this case V beta 11-2. And from this study from Moshe Arditi's lab at UCLA, they've shown that there's a increased V beta skewing in relation to MIS-C severity. They were also the first group to model this superantigen, showing it to be in the present in the spike protein of the SARS-CoV-2. So this is a really interesting, I would say, smoking gun when it comes to the pathology or pathogenesis of MIS-C. And to further support this, Lael Jonker, together with Moshe Arditi and other colleagues, were able to show that by SARS-CoV-2 present in the viral reservoir in the gut, eventually tight junctions in the epithelium of the gut will be broken apart, and S1 spike protein that is shed leaks into the bloodstream and can activate these T cells in a non-specific way through this super antigen mediated activation. And this presence of virus for a long time in the gut is probably a very important feature of MIS-C pathogenesis and could explain why the disease occurs with 1 month's delay from the initial infection, potentially, which is one of the really puzzling features of this disease. It's not only T cells that are activated, because when the antigen S1 leaks through the gut barrier, it can also form immune complexes with antibodies and trigger neutrophil activation and some of these other coagulopathy that is also associated with MIS-C. So this is intriguing. It's also important to point out that gut is really a central organ in MIS-C, and most of the patients do have some symptoms coming from the intestine. So, well, what about viral persistence? What do we know about this? And I think this is another area of SARS-CoV-2 infections that requires a lot more study. But basically, there is growing amounts of evidence now suggesting that viruses persist. And it's not only in patients with very severe acute COVID. For example, here's a girl, 11-year-old, with persistent intestinal pain more than 3 months after a mild COVID. And in a biopsy, viruses could be found and visualized. And a second study showing an adult, pretty young adult, with persistent SARS-CoV-2 present in the intestine, even 4 months up to 6 months after infection. And in this particular patient, there was also evidence of ongoing somatic hypermutation of the B cell receptors, which suggest ongoing continuous antigen stimulation. So I think these are just 2 examples. There are a number of other studies. There's a study of IBD patients showing that patients with persistent symptoms had presence of virus for a long time. So this brings me to sort of a hypothesis of why children typically, why is MIS-C predominantly occurring in children? Even though it's a rare condition, it's still sort of mostly in children. And one of the factors that I think is intriguing is that if we look at viral clearance in the nasopharynx, it's similar in both adults and children. But there is some evidence for a number of case series, at least, suggesting that viral shedding in the stool could be longer in children than in adults. So this supports this idea that there is something potentially related to the predominantly mild COVID in children and viral persistence. So what could that be? And so in the end of 2021 or early this year, I wrote this hypothesis in a perspective paper in Immunity, which basically states that one possible explanation for viral persistence and the dominance of MIS-C in children could be that children are more prone to disease tolerance versus resistance upon SARS-CoV-2 infection. And what do I mean by that? Well, what I mean is that in the face of an infection that is dealt with very efficiently locally in the mucosa, as we have already discussed, children might hold off on some of the additional activation of their immune cells and immune system in order to save energy and not to sort of waste energy on a very costly cytokine storm, or at least a sort of systemic inflammatory response, which is a very costly adventure, if you will. And we know that this is true for other infections. For example, EBV, primary EBV infections is typically asymptomatic or very mildly symptomatic in young children, whilst in teenagers, infectious mono can be very severe. And in those cases, we know that the main difference between primary infection in young children versus teenagers is bystander T cell activation, cytokine release, which could in turn, sort of in the case of SARS-CoV-2, also impact the likelihood of viral persistence versus clearance. And so that's the idea, that children that are growing might be more likely to withhold some of their energy from systemic inflammatory responses, which is a highly costly activity, whilst adults and older adolescents and so on might be more likely to elicit a stronger inflammatory response, leading to bystander T cell activation and potentially more effective clearance. So there is some support for this. For example, if we look at the energy requirement over the course of childhood, it's higher in the early years and then decreases with time. And so does the fraction of asymptomatic children, highest early on and then decreases with age. And MIS-C incidence also decreases with age. And intriguingly, there is a difference between boys and girls, both with energy requirement over the course of childhood, but also when it comes to MIS-C incidence, which is slightly higher in boys and sort of in keeping with this hypothesis. But I do want to stress that this is just a model and a hypothesis. There's very little data supporting this at this moment. All right, so what about hepatitis? We've probably all sort of quite concerned and puzzled by this new phenomenon where increasing numbers, at least in phases in different countries, of children under the age of 4 typically develop a severe acute hepatitis. And there's very intense, obviously, investigation ongoing all over the world to try to understand what's going on. And so far, the most sort of frequent finding has been an adenovirus infection of a adenovirus with a intestinal trophism, F41. But intriguingly, this adenovirus normally does not lead to hepatitis. When you're looking into the biopsy material of these livers, some of which have been transplanted, the hepatocytes are not directly infected with the adenovirus. So it seems like the hepatitis is not directly caused by the adenovirus infection. The second most frequent finding in these patients is SARS-CoV-2. Now, obviously, the first thing to do is to rule out any other cause, such as hepatitis virus A to E, or toxins, drugs, and other things. And in most of these patients, those things are not found. So this leaves us with a possible co-infection of SARS-CoV-2 and adenovirus. And I want to remind you of what I mentioned earlier from Miss C, which is that SARS-CoV-2 in the form of viral reservoirs can reside in the intestine of children for a very long time, 6 months, maybe longer. And that is not necessarily associated with SARS-CoV-2 positivity in stool. So one hypothesis could be that SARS-CoV-2 viral reservoirs and spike proteins, which have the superantigenic motif, could be present in these children when they encounter this adenovirus infection. And potentially then, the adenovirus infection could trigger the release of the superantigen and lead to a superantigen-mediated hepatitis. So is there any evidence of this? Well, there's some previous studies. For example, this is a mouse study from 2005 showing that mice that are sensitized with an adenovirus and then given SIC-BC superantigen develops hepatitis. And why do they do this? Well, they do this because the adenovirus infection polarizes the immune response towards gamma interferon and Th1. And then when the SIC-BC superantigen comes in, this leads to a much more severe inflammatory response. And because the adenovirus in this case is intestinal introphism, it leads to hepatitis. So we were intrigued by this, me and Moshe Arditi, and we wrote up this paper in Lancet Hepatology and Gastroenterology to urge our colleagues to start looking for evidence of superantigen-mediated T-cell activation in patients with severe acute hepatitis. Here's a second case where this was suggested to be an autoimmune hepatitis, but when looking at the livers, there's massive T-cell infiltration, and this patient recovers after steroid treatment. Potentially just a normal autoimmune hepatitis, but potentially also a superantigen-mediated disease. So what about long COVID? I'm kind of running out of time, so I'm going to speed up a little bit. But first of all, long COVID is a condition that we poorly understand. We really need to understand it much better. One of the things that I find clear is that the number of individuals with an objective measure of disease, meaning something that we can measure with existing tests, is a much smaller fraction than all of the individuals who have long-term symptoms, and particularly in children. But there are a few cases of children with severe lung damage after even a mild COVID. And we and others are currently investigating this. And the main hypothesis we are following up is that, again, this might be a form of superantigen-mediated disease, SARS-CoV-2 caused, and non-mutually exclusive, a form of autoimmunity, such as in the case of POTS, postural orthostatic tachycardia syndrome, where we know autoantibodies are the culprit. And all of this could be explained, at least in part, by viral persistence. So we are still looking into this. We have studied autoantibodies to interferons and find nothing in patients with long COVID. We're currently looking at immune dysregulation and so on. So with this, I'm going to end my talk and be happy to hand over the floor here. And these are just the members of my team that I'm very happy to work with. I'm happy to take questions towards the end here. So I'll stop sharing.
Thanks, Peter. That's great. That's a lot of ground that you've covered in a short space of time, and a lot of really great work that's being done. I think maybe we'll have 5 minutes now for any questions, and then we'll do Fatima's presentation, and then we can come back for more questions for both of you at the end. So either put your hand up, or you can type questions into the chat. I'm just gonna. There's one question early on in the presentation from Stephen about you showed that graph of increasing hospitalization rate over time. And the question is how that relates to increased adoption of vaccination. I don't know, Stephen, if you want to embellish or clarify the question, so I'm asking it correctly.
Right. No, you're asking it right. I guess I was more interested in that big gap with children in particular that seem to now, we're seeing more cases of severe COVID. And I'm wondering, does that correlate when you examine it carefully with the vaccination programs? Because early on, the children weren't as obviously affected, and now they are. And
yeah. It's a good point. I mean, the graphs that I was showing, they were all sort of divided. They were all based on pre-vaccination data. So and you also have to keep in mind that those were based largely on the 2 first waves and the variants that were present then, and not necessarily representative of the recent waves, right? When it comes to vaccination, obviously that's going to skew everything in terms of hospitalization rates, because vaccinations so clearly protect against hospitalization and severe disease across the entire age span.
Right.
So I think those curves I was basically showing just to illustrate the gradual increase in the pre-vaccination scenario. And I think it doesn't hold true now.
And I was curious, that super antigen that's in the spike protein, do you have the location more precisely?
Yeah, so it's right in the cleavage site of the PRR, the furin cleavage site. And so what's really puzzling about this is that if you just take that protein and you throw it onto PBMCs, so you would expect them to, the super antigen to work. It doesn't. And this has been one source of a lot of criticism around this theory. But the problem is that it's really difficult to produce the super antigen with intact 3-dimensional structure because it's so reactogenic, without adding, tags and other things, and those things mess up the structure. So it's really difficult to study this super antigen motif as it is. And so what we think, me and Moshe Arditi in the LA, who's sort of modeled this, we're trying to get a crystal structure going, obviously, but is that it's very conformational sensitive, and that's why it becomes difficult to study. But the TCR data is very clear and reproduced by 6 different groups. And so it's hard to explain that with anything else than a super antigen mediated activation, I think.
Yeah, well, thank you. It's very nice work.
Thanks, Peter. Megan, you had a question.
Yeah, thanks so much, Peter, for the fantastic talk. I was wondering, going back to the age-related effects of the disease, if in addition to the difference in, that you were talking about, the difference in metabolic requirements, if anyone's looked at the development of regulatory T cells, so as part of a normal counteractive response to all that inflammation, do you think that might be stronger in children and then gradually waning over time? Has anyone looked at that?
Yeah, so this is kind of one of my favorite topics, actually, and so I'm glad you're bringing this up. We study a lot of immune development. That's one of the things my lab has done the most prior to COVID. And what we see is that, yes, you have lots of Tregs during the very first months of life. In the blood, they sort of decrease quite quickly, and I think it's because they go to the tissues where they are needed, right? And so there is probably a more tolerogenic environment in young children, but that is, in my understanding, quite limited to the very first months and not something that will go on for 10 years or so. So I think it cannot really explain these protection that you see even in 5-10-year-old kids.
Okay, interesting. Thank you.
Thanks. So I think there's a couple more questions there. I think we will get to them, but I want to make sure Fatima has enough time for her talk, and
Sure.
So we'll come back to those at the end as well. So Fatima, if you want to start sharing your screen, and then I can start introducing you. So Fatima Yaseen is a research assistant at the Brockman-Brum Lab at Simon Fraser University here in BC, and her work primarily consists of analyzing antibody responses against COVID-19 and its variants from vaccination using the mesoscale discovery assay, which is an ELISA type assay. She's in the 4th year of a molecular biology and biochemistry and chemistry joint major at Simon Fraser, and hopefully will be applying to medical school in the upcoming cycle. So Fatima, over to you.
Thank you. Hi, everyone. Thank you, Dr. Brodin, for your amazing presentation. No pressure for me. But yeah, so today I'll be discussing some results from my research at the Brockman-Brum Lab. This data set that I'll be discussing is from a study that has a lot of collaborators, and there's already papers and preprints published with this work. So I would like to thank them all, and especially all the participants without whom the research would not be possible. One of the papers focuses on the magnitude and durability of humoral immune responses to COVID-19 mRNA vaccines among older adults. And here I've used the same data set and tested those plasma samples against a panel of variants to determine the ability of COVID-19 vaccines to elicit antibodies against variants of concern. So every virus purposefully mutates itself over time, leading to the emergence of variants. These variants are only of concern if they increase transmissibility, infectivity, change disease progression, or decrease effectiveness of public health measures, which includes vaccines. The variants of concern for SARS-CoV-2 as classified by WHO are Delta and Omicron. However, when I was collecting the data a couple of months ago, Alpha, Beta, and Gamma were also included in this list. And recently, Omicron has replaced Delta as the most widespread variant, with many subtypes emerging. The way that this was analyzed was using the mesoscale diagnostic instrument. We purchased plates that have up to 10 antigens stuck to the bottom of the wells. In our case, these are the S1 RBDs of the variants. The panels we purchased for these results is panel 22, and it has the following spots. The following data will show 2 subsets of antibodies produced by vaccination, binding and neutralizing. Binding antibodies will bind to the virus and can do many things from there to stop infection. One of them is neutralization. So neutralizing antibodies are a subset of binding antibodies that will disallow the virus from binding to the ACE2 receptor. The binding antibodies are reported as the log of the concentration, whereas the neutralizing antibodies are reported as percent ACE2 displacement. The plates have up to the plasma samples that are added are 1 to 10,000 for IgG binding and 1 to 20 for the ACE2 competition assay, which is a surrogate neutralization assay, after which a sulfo-tagged protein is added to label the antigen-bound antibodies for detection. Once the detection antibody is added, the plate is read by the instrument using electrochemiluminescence, and the signal is then analyzed by the software and back calculated to a standard curve to give a concentration in terms of AU per ml. The data set shown is from 3 time points: 1 month after the second dose, 6 months after the second dose, which allows us to determine durability, and 1 month after the 3rd booster dose. The cohort includes healthcare workers, long-term care residents, and seniors living in the community, and the age range is 24 to 98 years. Due to the cohort, the first thing to examine was the effective age on the vaccine responses. So the responses shown here are only against the ancestral Wuhan strain. And they show that 1 and 6 months after the second dose, the response of seniors and residents was significantly lower than the younger healthcare workers, while also showing on the right the responses of convalescents being the highest at their respective time points. However, 1 month after the booster, we can see the difference between the long-term care and healthcare workers is not statistically significant, providing some rationality behind administering the third dose, especially for the older adults. For ACE2 displacement at B1, the responses for older adults is still significantly lower, albeit to a lesser magnitude than at previous time points. When comparing the variants, the responses are not separated into healthcare worker and LTC, as the trends among the variants within these groups is the same. And as well, the convalescents have been removed from these figures. The responses of the variants was compared to the wild-type Wuhan strain at each of the time points, and a Mann-Whitney t-test was done to determine significance. So if p-value was less than 0.05, it was termed significant. So at 1 month after the second dose, binding antibodies for beta, gamma, and Omicron are significantly lower than wild type. 6 months after the second dose, all of the responses substantially decline. This supports the need for a third dose, and binding is lower for all except alpha. 1 month after the booster dose, all responses increase significantly to new peaks, and binding antibodies are lower against all when compared to Wuhan. The neutralizing responses are a bit different. 1 and 6 months after the second dose, neutralizing antibodies are lower for all variants when compared to wild type, whereas 1 month after the third dose, neutralizing is lower against all except the Delta variant. To conclude, the presence and the increase in responses after the booster indicates that the vaccines do elicit immune responses to all variants of concern. We can also see that 6 months after the second dose, there is a substantial decline in responses, and the booster also helps to increase that to a new peak. Antibody responses in older adults were weaker when compared to younger adults after the second dose, but similar after the third. The data also shows that particularly beta, gamma, and Omicron variants have mutations that may be immune evasive, as they have the lowest responses throughout. To further characterize the durability and strength of the response, it's important to consider the memory components of the immune system, which is something the lab is working towards. So I'd like to acknowledge all people from the lab and the study participants. Thank you.
Thanks so much, Fatima, for coming and sharing all of your data with us, and that's really exciting to see that work moving forward. So again, if people have any more questions for Peter or questions for Fatima, if you just put your hand up or post them in the chat. For now, I'll just keep going with some of the previous questions that people had put in for your talk, Peter. So the next one was something that I've heard raised by a few people around the acute hepatitis, and whether you think we'll see differences in people who are vaccinated with adenovirus-based COVID vaccines versus RNA-based vaccines?
Yeah, that's a really interesting question. I really don't know. I mean, the thing is, if this hypothesis actually depends on an adenovirus with intestinal trophism, then those, 80, as you know very well, I mean, 80% of those infections happen in kids under 4 years, right? So it's very young kids typically. And very few, I think very few such children have been vaccinated with adenovirus-based vaccines, at least to my knowledge. I mean, I don't know how anyway, to my knowledge, very few kids. So the likelihood of finding individuals to study there might be challenging. With that said, there might not be a complete reliance on adenovirus for this to work. I think potentially many viruses could potentiate a super antigen, and there's a lot of evidence for that from flu and other diseases. And so,
Yeah, no, definitely. I think the trials of those adenovirus in kids are ongoing. I don't think it's been approved in any country yet for children. And also, they're chimpanzee based. And I know in chimpanzees there's data showing that they have some tropism for the gut, but I don't know in humans whether it would be the same or not. The next question around, do you see the TCR-V beta repertoire analysis as being something that could be useful in assessing suspected MIS-C patients, possibly in patients also with the severe hepatitis, as part of their diagnostic evaluation? So is this something that could be translated into the clinic?
Yeah, so that's a great question. And Some of my colleagues here at Imperial, Mike Levine and his team and large group of people have been looking heavily at thousands of patients with various febrile illnesses and looking at transcriptome signatures. And they have a gene signature that distinguishes Kawasaki from MIS-C and a few other illnesses, toxic shock, very effectively. And I do believe that the TCR-V beta is actually one component in that signature. So I do think yes, probably. However, it's important to note that we're looking at the blood in these cases, and sometimes these super antigen mediated effects could be very local in the tissue. With respect to the hepatitis, we're currently right now sequencing some liver specimens and blood samples from young children to try to look at exactly this, whether we can see evidence of TCR skewing or not. We don't have the data yet.
Nice.
Okay.
Yeah. Be nice to have something specific that could help guide management early on, especially for those hepatitis patients.
I agree, absolutely.
Thanks, Catherine. So Fatima, there's a question here for you. So the participants in the data that you presented, are they all assumed to be not infected, or do you have evidence on their infection status either through antibody or testing?
So the participants that were in the variants, all of them are not infected up to that time point. They're all anti-N negative, but the one with the Wuhan, the convalescents were separated on that slide.
Okay, but Agatha, does that answer your question, or did you have?
Yeah, really simple. It's just I'm sort of aware of the cohort, and I think we knew there were exposures, and that always muddies the water. So it's nice that you separated those out. So sorry, perfect.
Sure. Stephen, you've had your hand up for a little while. I don't know who your question is for, so I'll let you unmute and ask it.
Yeah, hi, it's for Fatima. And I guess it carries on from the previous question because I was curious about that too. And I know with our work where we've been looking at the various proteins in the virus in terms of their immunogenicity, we actually find that the receptor binding domain region is very poorly immunogenic. And surprisingly, we find that the region where the nucleocapsid protein, about half the people that are PCR confirmed as having COVID, had the symptoms, don't have any antibodies against the nucleocapsid protein. So I think relying on a single marker for a non-spike protein as representative of actually not being prior exposed to the virus and having some immunity from that, I don't think you can rely on that. And I was curious also, Fatima, in terms of the response, now that some of this work has been published, you must be looking at people that have had their fourth shot, because in Israel there seemed to be losing that protection after a short while, around 3 to 4 months. Are you monitoring that, or have you seen anything like that yet?
So for the first question, I think it's just, we just use the anti-N Roche results to determine, and sometimes it's self-reported as well, like if somebody's had a COVID test, and that's what we use to exclude those people. And then looking at after the booster, we haven't examined any of the fourth doses, but we have some responses 3 months after the booster and now 6 months after the booster. And there is waning of the responses, but it seems to be at a lesser extent because the booster peak is quite higher than what we were at visit 3. So even going down from that 3 months later, we're still higher, a little bit higher than we were at visit 3. So.
And have you seen anything with children that are, under 12? Because the data from like New York State and from Denver that the FDA published seems to show that it's much less efficacious, the production of antibodies. Do you see that, or have you looked at that?
We have not looked at that because the cohort only has, I think 24 was the youngest participant that we had. So unfortunately, that's something that we didn't see.
Okay. Thank you.
Thank you.
So Stephen, we have a pediatric cohort that we've been following, so hopefully we'll be able to have some data from that soon. Jamie, there's a question, I think it's for you, Peter. What's your take on the increased severity of COVID-19 in kids with autoimmune diseases like type 1 diabetes?
Yeah, I saw that question. I don't know. I think that's quite difficult to answer to. I mean, in obesity and sort of metabolic disorders, it's possible that obviously that this idea of a energy allocation would be kind of derailed, and that people with obesity might be more likely to mount a stronger systemic inflammatory response. With type 1 diabetes, I mean, I'm not quite sure whether, I think one would have to take into account how well regulated are their metabolism, which varies a lot, obviously, between type 1 diabetics. So I don't have any good answer. I can only speculate on that.
No, fair enough. And then I think we've got time for one last question for you, Peter. So have you considered differences in responses between young and old could be prior exposure of older people to other human coronaviruses, and could cross-reactive pre-existing coronavirus-specific T cells from these previous infections also explain the super antigen effect rather than spike acting as a classical super antigen?
Yeah, I read that question and thought about it for a minute. I really don't understand how that would work, to be honest. I mean, first of all, absolutely pre-existing immunity could differ between different people, different ages. However, these, non-SARS-CoV-2, I mean, other coronaviruses are usually seen quite early in life. So most children would have seen them. I don't really understand how an immune individual would be more likely to mount a hyperinflammatory response, if you will. It's almost like an antibody-dependent enhancement, but on the T cell side, and I really don't quite understand how that would work.
Yeah, I don't know, Wolf, if you're able to unmute, if you wanted to clarify the question at all, or add a further comment.
Sure. So we've checked this, and others have checked it, of course, but there's lots of data that says pre-existing immune responses against other coronaviruses are predictive of convalescence after infection. So there's a reduction in hospitalization after infection, and there's a lot of data that says that there's a protective effect if you've made a response against one of the 5 to 8 other human coronavirus.
Sure.
So now you have an expansion of T cells, which are sharing the same antigen, because there's shared antigens between the coronavirus. Right now, you stimulate them, and I'm wondering if in some patients there's a predisposition to expanding those cells, which are now producing interferon gammas, etc.
I see. Okay. Yeah, no, I think that's a great point. I understand it better now. You're right, of course, that people who would have, in general, the super antigen will not trigger any T cell. It will trigger specific T cells, and that's why you see this expansion of V beta 11-2. There's also some potential links to HLA, which might be explaining why it's so rare. And so, yes, maybe you're right that having a lot of these cells expanded might make you more susceptible to this type of a response, potentially.
Thanks. I think just in interest of time, we just need to start wrapping things up. So just firstly, a huge thanks both to Peter, who I think when we first planned this, he was planning to be in Calgary, but he's still in the UK, so it's sort of late evening for you there. So thank you so much for joining us in the evening your time, and thank you, Fatima, also for sharing your data with us as well. Thank you so much, everyone. Thanks.